Hello, everyone. Good afternoon. I'm Debbie Hart, the President and CEO of BioNJ, the Life Sciences Trade Association for New Jersey. I'd like to thank you for joining us for Positioning Early Cell Therapy for Clinical Trial Success, Mitigating Risk, and Ensuring GMP Readiness, brought to you in partnership with BioNJ member Thermo Fisher Scientific. As we know, the promise of cell therapies is often tempered by the many complexities of transitioning these innovations from R&D to the clinic. With this in mind, our presenters will dive into a discussion on how to navigate the evolving cell therapy regulatory landscape, assess GMP readiness, and prepare your cell therapy workflow for clinical and commercial manufacturing, and ensure patient centricity in the complex cell therapy ecosystem and leverage a unique, comprehensive operating model for your product. So today's webinar is being recorded and will be available tomorrow at bionj.org. We highly encourage you to ask questions by placing them in the Q&A box. And now let's get started. Please welcome from Thermo Fisher Scientific, Senior Manager of Regulatory Affairs, Michelle Dugan, Director of Cell Therapy Sciences, Dr. Mindy Sadik, and filling in for Dr. Kim Watanabe, who unfortunately was unable to join us today, is Senior Director, Project Delivery, Hematology Oncology at PPD, the Clinical Research Business of Thermo Fisher Scientific, Dr. Vito Romita. And Vito, thank you so much for stepping in at last minute. This is a terrific group and look forward to what you all have to say. So welcome to all of you, and the floor is yours.
Well, thank you for the introductions, and hello to everyone joining the webinar today. In this first segment, I will be sharing my thoughts on navigating an evolving cell and gene therapy regulatory landscape. I'll be looking at this topic from a U.S. regulatory perspective for sake of time, but the approaches discussed within this presentation are common to other regulatory regions. Next slide, please. So when you look at the regulatory submission activity in the cell and gene therapy space, you can see from the graph a significant rise in IND submissions to FDA over the years, with original IND submissions nearly doubling from 2017 to 2021 when compared to 5 years prior. Further, the FDA predicted in 2019 that there will be 10 to 20 new cell and gene commercial therapies approved annually by 2025, which does present an intriguing landscape for innovators, but also for the regulator and how they address the increase in workload that they will encounter. The Alliance for Regenerative Medicine, or ARM, summarized FDA's decisions in recent sector snapshots. And as we approach 2025, we see that FDA may not have been too far off in their prediction. A total of 6 market applications were approved in 2023. And this current year is shaping up to eclipse last year's with 5 market approvals so far to date, and possibly for another 4 based on the number of BLAs submitted in 2023. Next slide. Let's take a moment to review aspects of the cell and gene therapy that could present real-life regulatory challenges for those that operate within this modality. The list on this slide is not meant to be an all-inclusive list of all challenges experienced, but do present as significant obstacles. Firstly, cell and gene therapy products have an inherent complexity. Unlike other modalities, these challenges encompass manufacturing and logistical complexities, while having to navigate multiple regulations, such as those related to GMP, biologic regulations, possibly device regulations, amongst others. FDA also offers several expedited programs, which puts many of these therapies potentially on an accelerated path, requiring innovators to plan early and be prepared. This translates into a need to plan early for scale-up, transfers, any process changes required to move towards and support early market entry on an accelerated schedule. Secondly, the rapidly evolving regulatory landscape presents challenges for the industry. Over the past decade, the FDA has released guidelines at a relatively slow pace, and it's really just been in the last year or two where we finally have an uptick in released guidelines. Also, based on the diverse nature of cell therapy products and technologies, it can be difficult to effectively interpret and apply FDA recommendations described in these guidelines. Finally, FDA workload. As presented on the last slide, there has been a tremendous submission activity for the regulator, whether it's at the IND stage or commercial, requiring more resources at FDA to handle the influx. This can have an impact on timely feedback, as well as sponsors receiving consistent advice. The ripple effect is potential impact to IND approvals in the form of clinical holds and/or delays in development timelines. Next slide. There have been several notable initiatives in the last few years by FDA to address the challenges presented by the growth in this market. In March 2023, CBER underwent a reorganization in its Advanced Therapy Review Office and established what's called the Super Office, or the Office of Therapeutic Products, or known by the acronym OTP. As you can see from the organizational chart, the new Super Office consists of 6 sub-offices, 14 divisions, and 33 branches. The new structure highlights the importance of manufacturing for OTP products, with 3 dedicated divisions focused on CMC, a division specific to gene therapies, another specific to cell therapies, and a third covering other products such as plasma proteins. So by aligning disciplines and product types, it's anticipated that the consistency in the regulatory advice provided and the consistency of submission reviews will be much improved. Further, the increase in resources will help FDA to be better equipped to address the continued growth in this sector. Next slide. Another initiative by CBER in 2022 was to host virtual town halls, really as a way to expand its outreach and offer another means to discuss topics related to OTP regulated products. To date, the town halls have rotated amongst the various review divisions, including CMC and clinical review teams, and separate town halls have been organized for gene therapy products and cell therapy products. The Q&A have covered topics that clarify expectations on specific technical components required for INDs and market applications. Topic areas have included common CMC issues leading to clinical holds, potency assay approaches, and also implementing and advocating for sponsors to implement changes early in a development program. Town hall transcripts and video recordings are available on the FDA website. OTP also has a Learn series, which lists webinars and courses presented by FDA staff on a variety of topic areas. So I would encourage this audience, if you're not familiar, to have a look at the OTP Learn series and workshop web pages. Next slide. So the FDA has progressed in issuing guidelines focused on targeted topics within the cell and gene therapy space. So the first cell therapy guideline was released back in 1998 and was followed nearly a decade later in 2008 by a more specific guidance on INDs. Fast forward to January 2020, FDA issued its first detailed CMC guideline for gene therapy IND submissions. But over the past year and a half, there has been a surge in activity with several important guidelines being released. One of the most highly anticipated was the manufacturing changes and comparability guideline that addresses the unique nature of cell and gene therapy products, which often have shortened development timelines and complex change management requirements. At the end of 2023, The FDA issued a draft guidance on potency testing, a hot topic for innovators, and how to apply a science and risk-based approach to potency assay development. The final guidance for CAR T cell products was issued earlier this year, containing specific recommendations for CMC, farm tox, clinical study design, and notably, it also addresses the FDA's perspective of the advantages and disadvantages of single versus multi-site manufacturing. In April of this year, the FDA also issued 2 new draft guidances for industry comment. These address the use of human and animal-derived materials in manufacturing cell and gene therapy products, and the safety testing of human allogenic cells expanded for use in cell therapies. So overall, there's been an increase in FDA guidance activity, providing much needed clarity and direction for this evolving field. Next slide. But there are often questions that come up during development that cannot be fully answered by guidance documents. There are a number of ways to engage FDA during development, as outlined in this graphic. The pre-IND meeting is an important milestone meeting for any program, providing an important step to establish a collaborative relationship with FDA reviewers before initiating clinical studies, and helps the review division become familiar with the product and process. However, for complex and innovative products, this single interaction may not be enough from a clinical and CMC perspective. CBER has provided additional touch points during early development to engage with FDA staff upstream in the development process and prior to the pre-IND, such as a CAT and interact opportunities. Once clinical trials are initiated, there are opportunities throughout clinical development to engage with FDA. For traditional INDs, milestone meetings like end of phase or pre-BLA, and specific topic meetings like Type C or the new Type D meetings can be utilized. For those products granted an expedited path, such as RMAT or breakthrough, these pathways offer perks of frequent interactions with FDA as part of their designations. Next slide. So as mentioned on the previous slide, there are 2 early engagement meeting types that can occur prior to the pre-IND. If a novel platform or technology has been developed, then early consultation for non-product specific questions can be requested through CBER's Advanced Technology Team, or CAT. Consultation is in the form of either written inquiry or a meeting request. An interact meeting is another example of an early engagement opportunity that provides informal, non-binding, one-time consultation with CBER for innovative products. This meeting is intended to be a very early product development discussion, seeking to facilitate feedback on novel or challenging issues that might otherwise delay entry into the clinic. Next slide. So meeting with FDA can present itself as a daunting task, but with the right preparation, maximum impact of the engagement opportunity can be realized, which is important when de-risking and accelerating a development program. Further, engaging your manufacturing partners in the meeting preparation process is an excellent opportunity to leverage their experience as a way to enhance agency discussion and feedback. So firstly, it's important for sponsors to ask well-focused, specific questions in order to produce actionable answers. For that reason, questions should not be open-ended nor broad in nature. Sponsors only have access to one pre-IND meeting, so it's important to seek clear and meaningful advice. Secondly, be prepared to ask the hard questions by evaluating and having a good understanding of potential gaps and issues in your program. These may be gaps or issues that maybe you're worried about, if addressed now, can potentially slow down development. But agency feedback enables realistic planning, and if sought early in the program, can avoid costly rework or even future delays during development. Also, develop a game plan, including contingency measures, in the event that the regulator does not agree with the company's position. This speaks to being prepared for any outcome. Finally, avoid ignoring advice. Not only does this potentially increase the risk for delays, but it can hinder the ability to foster successful collaborative relationships with the regulator. If clarifications are necessary, then ask for them. If a different approach is being taken after engaging with the regulator, then discuss the rationale for doing so in a future advice meeting or within the regulatory submission itself. Next slide, please. So in closing, the regulatory landscape has rapidly evolved and continues to do so. And there has been important steps taken by FDA in recent years to provide clarity to industry on regulatory matters, as well as to increase their own resources so as to facilitate the development of cell and gene therapy products. This increase in regulatory activity requires all stakeholders in this sector to continuously monitor and stay informed on regulatory trends and guideline changes. Recognize those opportunities to meet with FDA during development, including opportunities early on to discuss and align on plans, challenges, and the path forward in order to de-risk development activities. And finally, there's tremendous value in leveraging strategic partnerships and knowledge sharing in order to improve the chances of clinical and commercial success. I will now hand the presentation over to Mindy for a discussion of preparing your cell therapy workflow for GMP compliance.
Thanks, Michelle. Next slide, please. So to start out, I wanted to discuss some of the common challenges that we see in cell therapy manufacturing. As Michelle just discussed, the ever-evolving regulatory landscape. So with rapid advancements in cell therapy technologies, regulatory bodies are continually adapting to ensure patient safety and efficacy. And as she described, navigating these regulations can be a significant challenge to manufacturers. This often requires extensive resources and expertise to remain compliant. The second challenge I wanted to discuss was the high cost of goods associated with cell therapy manufacturing. So the manufacturing process is often complex and very labor intensive, requires specialized materials, reagents, specialized equipment, skilled personnel, and stringent quality control measures. And all of this contributes to a higher production cost for the final drug product. Labor-intensive processes represent yet another obstacle in cell therapy manufacturing, especially since it's really difficult to standardize workflows across programs. Many aspects of the production will require manual intervention, which can increase the risk of error and variability. The lack of optimized analytics also presents a significant challenge in assessing product quality and performance. Because cell therapies are inherently complex biological products composed of living cells, they of course exhibit heterogeneity in terms of their phenotype, potency, and behavior. And therefore, traditional analytical methods might not be well suited to accurately characterize and assess the quality attributes of the cell therapies. So this too can lead to some challenges in product characterization, process monitoring, and quality control. Achieving commercial scale capabilities can also be quite a challenge when a manufacturing process needs to be scaled up to manufacture something like thousands of doses. So identifying a scalable manufacturing platform, optimizing process parameters, and validating the manufacturing process to ensure consistency, reproducibility, and compliance with all of the regulatory requirements is extremely time-consuming and resource-intensive. Workforce development and retention has also become quite a challenge in cell therapy manufacturing. This is due to the shortage in skilled operators and scientists, and unfortunately also due to the shortage of training programs in this area. So as the field continues to grow, there's a growing demand for these skilled professionals. And then last but not least, a complex supply chain. Coordinating the procurement of raw materials, navigating import-export regulations, and managing distribution logistics requires careful coordination and collaboration with the suppliers, the partners, and even the regulatory authorities. So now that we've talked about some of the challenges, I'd like to spend some time talking about considerations that should be taken to achieve manufacturing success. So next slide, please. Great. So these considerations on this slide are really essential to ensure product quality, consistency, and regulatory compliance throughout the entire manufacturing process. The first point I'd like to make is around raw material selection and management. It's important to ensure that your raw materials meet your specifications and that they can be used for GMP purposes. It's also important that when they arrive at the manufacturing facility, that they're received correctly, documented correctly, and brought into the supply chain correctly. This includes storage conditions, temperature controls, etc. Whenever possible, early on, try to choose GMP over research use only materials. So this includes everything in your process, your reagents, your media, your supplements, your cytokines, serum, etc. Otherwise, as the clinical trials move forward, comparison studies will need to happen, and this will add time and cost to your process. It's really also important when you're choosing materials for a process to remember to have dual vendor sourcing and that these vendors are in your CMC when you file your IND, so that if there are any issues of not being able to receive materials on time or having some sort of supply chain issue, you can pretty easily switch to a second vendor and can also help save you time and cost. Another really big consideration when developing a workflow is to reduce as much manual touch points, wherever possible in the process. So this means employing closed systems, automation, and even single-use disposable materials if possible. Obviously, one of the greatest benefits of using a closed system is that you can reduce the risk of contamination through exposure. And avoiding contamination is not only important for safety reasons, but also contamination can lead to a high cost with product loss and cleaning. Open cell manufacturing systems really require experienced professionals to conduct the steps in the process. And closed systems, on the other hand, don't really rely as much on an operator. Therefore, they're less prone to human error. In addition, the full automation of a process eliminates extra steps, making the entire process faster. The next consideration is scalability. Scalability really ensures the ability to meet increasing demand without compromising on product quality or efficiency. And so a key consideration when developing your cell therapy process is to implement production platforms that are scalable. Depending on the dose required and the number of patients that will be treated, the proper scalable platforms, so things like bioreactors or cell harvesting methods, really should be determined in the early stages of product development. Another consideration is manufacturing records. So you want to ensure that you have a detailed protocol for your manufacturing process, and that the master batch records are in a place that are really documenting the process and all of the activities. Thorough documentation is crucial for regulatory compliance. Optimized analytics are yet another consideration. So it's important to develop robust and qualifiable assays, as materials coming out of your GMP facility will have to go through a quality control lab for testing. This means leveraging advanced technologies to really build process understanding. Without reliable analytical methods, manufacturers may struggle to ensure consistency, reproducibility, and scalability of their manufacturing process, all leading to batch-to-batch variability and, unfortunately, product deviations. So ensure that your analytical methods are developed appropriately and that are developed on platforms that can be translated to a QC space. Also ensure that they have the proper standards and the controls. Many traditional analytical techniques, such as flow cytometry, ELISA, PCR, etc., have some limitations when applied to cell therapy products. So it's really important to incorporate new and emerging technologies like single cell analysis, omics technologies, advanced imaging technologies, etc., into your workflows. Quality systems. Design phase appropriate quality oversight into your cell therapy program. So this means implementing quality systems that are really tailored to the specific phase of development that you're in, from early preclinical research throughout clinical trials and finally through to commercial manufacturing to ensure you're compliant and your product is safe. Quality systems help to identify and assess and mitigate any risks associated with the manufacturing process. And by implementing phase-appropriate quality systems, manufacturers can really prioritize risk management activities based on the stage of the product development, potential impact on patient safety, and regulatory expectations. And then lastly, quality control release testing. Ensure the final product meets the basic safety requirements and adheres to the release specification that's set out in your protocol and that's approved. So next slide, please. So at Thermo Fisher, we like to say begin with the end in mind. Developing a cell therapy workflow for GMP manufacturing is a very complex and multifaceted endeavor, and it requires careful planning, strategic foresight, and meticulous attention to detail. Start early, begin with the end in mind. These are foundational principles that can significantly impact the success of your manufacturing process. We're really lucky again at Thermo Fisher. We have a team called our Translational Services team. The idea of this team really is to support our GMP destined customers early, and therefore they help allow for a seamless transition of a process and analytics into our GMP facility. By optimizing your process, even at that early preclinical stage, you can lay the groundwork for a scalable, reproducible manufacturing process in GMP. And by working with a team like our translational team, for example, developers have access to many different technologies, lots of different platforms, equipment, and even automated solutions that can help inform decision-making and strategic investments for your manufacturing efficiency. This team also helps developers identify GMP equivalents to any research use only materials. They offer scale down processing models that can mimic large scale manufacturing conditions. And they perform in process testing and monitoring to really better characterize and optimize processes. They stay in close communication with our regulatory experts, as Michelle described, to ensure that they're developing methods based on the latest guidances. And it's really important when designing your manufacturing process that you have that tech transfer in mind to facilitate a seamless transfer and accelerate time to market. So next slide, please. So at Thermo Fisher, we have a comprehensive way that we will assess manufacturing workflows and make sure that they're ready for GMP manufacturing. First, we conduct what we call verification runs to perform manufacturing process in our facility, on our equipment, and with our personnel. And by looking at this and performing these runs, the team can identify any risks and start to formulate any gaps and optimization studies or solutions that can help eliminate risk and ensure process robustness and reproducibility in GMP. We look for open steps in unit operations and we devise ways to close them. We take manual operations and we see if we can introduce automation. And so by streamlining and optimizing your manufacturing process, it can really help to reduce production costs, save time, improve efficiency, minimize waste, and increase yield. On the bottom of the slide there, you see some of the areas of process development that we really tell developers to focus on and that we can help with: process optimization, scale up and capacity modeling, process and product characterization, closing and automating steps, as I mentioned, helping with fill finish activities, cryopreservation, and finally performing comparability and stability studies. And by implementing a really strong, comprehensive process development strategy, cell therapy manufacturers can expedite that transition from preclinical to clinical and commercial manufacturing. And this proactive approach not only accelerates time to market, but enhances process robustness, quality, and ultimately patient access to these life-saving therapies. Next slide, please. So this slide speaks to the analytical development strategy and services we offer. As I mentioned, analytical development for cell therapies is a critical aspect of the manufacturing process aimed at ensuring safety, identity, purity, potency, and stability of the starting material, in-process material, or the final product. And at Thermo Fisher, we have a really stringent workflow we follow to ensure we develop robust, compliant analytical methods that, again, can be transferred into our quality control space. So for cell therapy products, it's essential to look at several critical assays, including safety, which is designed really to assess the absence of any contaminants, including microbiological contaminants, mycoplasma, endotoxins. So for example, we use rapid sterility testing. We use a PCR-based mycoseq approach for mycoplasma testing, and then we use EndoSafe for endotoxin testing. We also perform identity assays to confirm the specific identity of the product and verify that it actually matches the intended cell type. So typically flow cytometry can be used to assess the expression of certain cell surface markers that, again, will confirm the identity of the product. We also perform purity assays to determine the level of impurities present in the cell therapy product. Typically, we use flow cytometry to evaluate percentage of target cells, so for example, CAR T cells, and to assess the presence of any unwanted cell populations. Cell counting is also used to quantify the number of viable cells in the product and assess the presence of any non-cellular components. Potency assays measure the biological activity of the cell therapy product and really demonstrate its ability to achieve the desired therapeutic effect. So things like cytotoxicity assays can be developed to measure the ability of the cell product to kill target cells, for example, cancer cells in vitro. Functional assays can also be used to assess the specific biological activity or function of a cell therapy product, such as cytokine secretion or immune cell activation. And then lastly, we perform stability assays to evaluate the product's stability over time. And we can do this under various storage conditions to make sure that the product is safe, efficacious, and suitable for clinical use throughout its shelf life. These analytical assays, as I mentioned, are essential to ensure safety, quality, potency, and stability of cell therapy products throughout the manufacturing process and prior to clinical administration. And by implementing robust analytical assays in your manufacturing, manufacturers can really comply with regulatory requirements, maintain that product consistency, and again, ensure product safety and efficacy. So last slide, please. So some closing thoughts, things I discussed. Begin with the end in mind. You want to do this to really reduce downstream risk and ensure, again, a more seamless transition into a GMP facility for clinical phases. Start early, plan for GMP manufacturing from the outset. Consider things like scalability, regulatory requirements, and supply chain management. Integrate advanced technologies into your workflow if possible to really optimize efficiency and mitigate any risk. This includes things like leveraging automation, using closed systems, and using real-time monitoring to really enhance efficiency and mitigate risks that are associated with manual processes. Develop qualifiable, transferable analytical assays that have the appropriate controls and standards. And by doing this, you'll really enable an accurate assessment of your product's quality and consistency. Assays should be designed with transferability to a QC team in mind and facilitating a seamless integration into routine quality control processes. Consider leveraging the support of an experienced CDMO partner when your expertise or capacity in-house is unavailable. And that can really provide you access to expertise, resources, and capacity, which will overall help accelerate development timelines, mitigate risk, and really optimize your manufacturing process. And by embracing these key principles, cell therapy developers really can enhance the likelihood of success in preparing their therapies for GMP manufacturing. And so with that, I'd like to turn it over to Vito to talk about ensuring patient centricity when conducting cell therapy clinical trials.
Hello, everyone. A pleasure to really address this audience today. and really speaking about patient centricity, and also tying in many of the topics that both Mindy and Michelle have brought to light during this very informative discussion. Next slide, please. So when we talk about the clinical trial landscape, I think what we have to also start to think about is all the processes that are or that we have to address in terms of how it impacts the patient experience. And so throughout my presentation today, I'll be touching on the various elements, considering all stakeholders, that is our customers, the developers themselves, the regulators, the payers, of course, the investigator sites and their patients. And throughout the discussion, I plan to touch upon some of the topics that are so critical to the success of any clinical trial, thinking about moving from the benchtop science to market access, capitalizing on many of the discussion points made to date, but relating these topics to boots on the ground operations. I think as a preface, we cannot really have an honest discussion on patient centricity without understanding some of the challenges along the development life cycle of a cell therapy asset and how we as collaborators in the industry can partner and address some of these challenges. Next slide, please. As part of Thermo Fisher, PPD as part of Thermo Fisher is well established to meet and address as well as support the industry along the entire life cycle of an asset development. PPD provides full CRO services to respond to these requirements. We provide scientific, medical, protocol design, regulatory, as well as project management expertise. We certainly provide an extensive and robust global framework with respect to managing the patient journey and the cell journey. Both Mindy had made reference to supply chain management, especially with respect to cell therapies when we think about cold and excuse me, ultra cold chain supply management. We also have to consider market access and how we apply effective strategies early on. And this, in addition to the wide range of analytical services supporting translational medicine, early and late stage development. And of course, we have to think about the patient services, including patient advisory groups and other patient support services to enhance that clinical trial experience for the patient in terms of understanding clinical trials, advocating for clinical trials, and participating in clinical trials. Under the Thermo Fisher umbrella, as both Mindy as Mindy has just mentioned, we provide a number of services that span the gamut of drug development, whether that be providing specialized equipment, analytical capabilities, genetic analyses, manufacturing of viral vectors and cells, and certainly all of the additional considerations around supply management, again, addressing some of the CMC and GMP considerations that are so important. And so the main message here, as we develop this conversation, is to think about what are the critical factors that can risk the development of an asset. Scientific robustness and regulatory implications. We certainly have had a little bit of a snapshot today with respect to the complexity of the regulatory landscape that is out there, where PPD, part of Thermo Fisher, can certainly support. We have to think about tech transfers in terms of product characterization, in terms of scalability. Think about moving from an investigator-led trial where the use of fresh cells is being utilized, and then scaling up to cryopreservation cells or cryopreserved product. That changes the landscape from a regulatory perspective, but also from a scientific perspective considerably. The ability to scale up manufacturing and ability to reduce manufacturing timelines. We know from conversations with patient advocacy groups as well as the patients themselves, that this is the root cause or central to patient hesitancy in participating on clinical or in clinical cell therapy trials. And so we need to be able to address scalability, timelines to production, and in addition, looking at the critical trial design itself, where there might be inherent biases in the inclusion-exclusion criteria, and how we will approach ensuring capturing of the relevant safety data, both short and long term, addressing limitations with respect to patient access to these potentially life transforming therapies, and how we can also capitalize on our patient services and decentralize clinical trial strategies, which also have significant regulatory implications, especially as we think about the application of those strategies outside of North America. So there's a lot to think about, and throughout this entire process, we have to leverage the expansive depth and the expertise and capabilities that is indeed a critical part of our value proposition, bringing in and solutioning many of these challenging elements as we aim to deliver a meaningful experience to the patients that participate. Next slide, please. Um, Again, to address Thermo Fisher's capabilities, we certainly have a tremendous track record. So, for example, we have over the last, at least through August of 2024, 49% of our FDA-approved cell and gene therapy products were supported by our CDMO and PPD CRO services. Next slide, please. This slide is only to, again, emphasize the scope and the depth and breadth of which PPD, part of Thermo Fisher, and partnering with Thermo Fisher, can bring these services, the expertise, the subject matter experts, so that we could better inform and collaborate with our partners and our stakeholders within the industry to provide and really execute an efficient clinical trial. Next slide, please. If you can just click on so that we can, thank you. So I want to take us back a little bit just in terms of translational medicine and making that distinctive switch from early discovery to initiated clinical trials, right, and then bringing that right through to IND filing and launching an early phase trial, and then of course expanding to the pivotal phase or commercial development, as well as post-approval. And I think what has been a common theme throughout the entire conversation today is the regulatory robustness, the ability to characterize the cell product, understanding the scale up of that product at the various GMP facilities to ensure that you have comparability, thinking about the robustness, the GMP required reagents and analytical assays with the appropriate controls so that we can make these clear-cut comparisons. And certainly we have to demonstrate the clinical evidence in terms of efficacy and safety. And so again, through the PPD services as part of Thermo Fisher, we can certainly address these very challenging aspects of clinical trial development, especially as we move from benchtop and into the clinical trial setting. Next slide, please. So with respect to the entire pathway, I think it's really important that we identify some of the critical aspects with respect to drug discovery and development. We know that it is an incredibly expensive endeavor, costing over $1 billion for each new drug or asset that is approved for clinical use. and this with no guarantee at the onset that there will be any successful outcome. And so throughout this process, we have to apply a very robust mindset so that we could tackle some of the challenging aspects of clinical trial development. We talked about scientific and clinical complexity, this around formulation, stability, efficacy, drug safety. We talked about process development and optimization, where we now have to deploy our technical technical strategy in terms of translational, moving from early clinical, preclinical, and then into clinical trials. Very often, we do not include or consider some of the regulatory requirements in terms of data capture with respect to the type of data, both long-term and short-term, so that we could satisfy the agencies, both in the FDA and in Europe, when we are at a point or a pivotal point where we're considering a marketing application. The operational complexities, Mindy made reference again to the supply chain, but we also have to think about deburdening the sites given the massive research and development in terms of investment expected through the end of this decade. There's a tremendous demand on site resources. And then from a patient experience, trying to understand what's expected of them, how are we going to make it possible for them to participate? Earlier on, I made a reference in terms of patient hesitancy. And certainly, accessibility, the timeline to manufacturing the product and receipt of that product, but also how do we manage the patient experience and deburden the patient in terms of accommodating travel, thinking about missing work, childcare. The list goes on, and this is another significant factor where enrollment and data collection based off of enrollment is impeded because of some of these considerations and concerns that really the patients, to use in layman's terms, do not or cannot get their head around. And so there must be a tremendous amount of support in terms of education from a patient onboarding perspective, as well as from a infrastructure and clinical design perspective, ensuring that we're collecting the right amount of data, the right data, so that we could have an easier time in commercializing the product. May I then go to the next slide, please? Just very briefly, the majority of our cell therapy experience, or at least predominantly, has been in the hematology and oncology space. Certainly with 6 approvals, FDA approvals for cell therapies directed towards the treatment of hematological malignancies. But we also know, as of very recent, there is a tremendous expansion in terms of where cell therapies is headed and directed, and this involves other non-hemonc indications, for example, in autoimmune. If you can please click to the next so that thank you, including autoimmune, cardiovascular, metabolic diseases. And so with this new chapter comes additional challenges. And I think the next slide, is going to be quite telling and informative to this audience, because it is a quite unique situation where, again, there needs to be a tremendous amount of consideration with respect to onboarding sites and patient education. Next slide, please. What this slide basically illustrates is a pathway for which a patient is diagnosed, for example, in an autoimmune indication. The primary care physician and the investigator will very likely be the rheumatologist, immunologist, the nephrologist. But these specialties are not as familiar with cell therapies. They're not as familiar with onboarding patients and educating these patients. They are not as familiar with the management of the safety events that are elicited by many of these gene-modified cell therapy products. But the hematologist oncologist is. So when we start to approach these sites in early identification or site identification, we have to consider the onboarding of these immunologists, rheumatologists, nephrologists, again, in the context of an autoimmune indication, and really connect the dots between the developer, the principal investigator, and of course, the hematologist oncologist who is experienced in managing patient onboarding, site selection, and providing close safety surveillance. There is another element with respect to the various steps along the clinical trial pathway when we're thinking about, excuse me, when we're thinking about onboarding these patients and onboarding the sites. So we talked about site selection, but also startup considerations. We talked about the patient selection and onboarding, as well as consenting and educating these patients on these complex trials, and providing an understanding of the potential risks. There is a coordination across several departments, locations, where apheresis, pharmacy, infusion centers, intensive care units, how will all this be managed from a startup perspective, from a budgeting perspective in terms of initiating clinical trial agreement negotiations. All of these play an important role in managing the patient journey as well as the site journey with respect to startup and patient onboarding. The negotiation of the slot assignments and the manufacturing. Again, this is a very complex area where you have to think of how we onboard these patients, collect the starting material, the cell starting material for the manufacturing of these of the cell product, and how do we then move that product again across the various sites across the various countries, so that we could again ensure that these patients have timely access to that product. Again, circling back to my first comment about patient hesitancy and their concern about the time it takes to access some of these treatments in terms of the waiting time for the treatment to be manufactured. Next slide, please. I'm going to finish very shortly by just saying that indeed there is a multidimensional clinical trial approach that we have to consider for cell therapies. We target the disease based off of prevalence, based off of incidence. We have to come up with an effective country and site mix. We certainly need to take into consideration many of the regulatory implications and complexities in terms of startup. Certainly drug design and trial design are important elements where we have to start considering the patient's voice, patient advocacy groups earlier on in developing these protocols. We have to consider inclusion and diversity. And certainly we have to consider the technologies to deburden the sites in terms of data entry. Patient centricity in terms of including or considering rather patient retention over the long run, where we have to collect safety data for up to 15 years post their last cell therapy treatment, where the trial designs for these long-term follow-ups are indeed interventional. And then, of course, as we discussed, manufacturing in terms of the supply chain, in terms of managing the patient journey and the cell journey, in terms of having a global and robust infrastructure to support those logistics. These are all important considerations that have to go into the trial design approach. The last slide, please. Again, to capitalize on both Michelle and Mindy's talking points, the one important thing where we combine the CDMO and CRO services together collaboratively under the auspices of PPD's Cell Therapy or Cell and Gene Therapy Institute, as well as our Cell Therapy Center of Excellence. We bring expertise right across all of the various business segments within PPD, from manufacturing, the regulatory approach and clinical strategy, and then the actual execution of those clinical trials, and then collecting real world evidence to inform us on strategies to get these products to market. So I will finish on that note, and thank you very much for this opportunity.
Terrific. Thank you so much to all of our speakers, Mindy, Michelle, and Vito. I think we have time probably for one question, it looks like. And Michelle, that's coming to you. So with all the recent efforts by FDA to increase resources and guidelines to the cell and gene therapy sector, is that signaling perhaps that FDA may be raising its expectations for sponsors?
So from my perspective, I don't necessarily view this effort as FDA raising expectations per se, but instead of finding ways to give sponsors more regulatory clarity and how to view and understand statutory requirements, this sector is diverse, evolving quickly. And as FDA learns, as much like the path taken for other mature modalities, this is FDA learning, translating it into guidance to help sponsors progress development.
Okay, thank you. Thank you very much. I'm checking the chat. I don't see any other questions. So with that, given that we are basically out of time, I would like to say thank you once again so much to our speakers. We very much appreciate your time and your expertise. If you do have a question as a follow-up, please email us here at BioNJ at bionj.org. We'll be happy to get them to the speakers and respond to your question. As a reminder, a recording of today's program will be available on our website tomorrow at bionj.org. We hope to see you at a future BioNJ event. And you can see all of our programs at bionj.org. And thank you so much to Thermo Fisher for partnering with us on this program. And Thermo Fisher is part of our purchasing consortium. And if you're not a member of BioNJ, we welcome you joining. Sherry and Danielle can help you. And finally, we have a QR code that will be on the screen in a moment, and we invite you to take our short program evaluation. Your feedback is very, very important to us. So thank you again to our speakers. Thank you to Thermo Fisher. Thank you to our BioNJ team who executed this flawlessly. And thank you all for joining us this afternoon. I hope you enjoy the rest of your day.